Cellular respiration takes place in a couple different locations inside the cell. So if we review the overall process of cellular respiration and we put those different stages inside the cell where they do occur, our first stage that we're going to be looking at is out here in the cytoplasm. So this is going to be glycolysis. What glycolysis is going to do in the grand scheme of things is that glycolysis is going to begin the breakdown. So it will begin breaking the glucose molecule apart. And if we actually just break the name up, if we break down the word glycolysis, it means splitting of the sugar, breaking the sugar. So that does really sum up what the process is going to do. It's going to begin the breakdown of the glucose. When it does that, it will make some ATP. Now, the amount of ATP that it makes is going to be pretty minor. It's about 2 ATPs, and that ATP is made by substrate level phosphorylation using a kinase enzyme. And then the other big thing that glycolysis is going to do is it is going to start to collect electrons from the glucose. So it will begin the collection of the high energy electrons. The molecule that will do the collecting is going to be NAD+. So NAD+ is going to grab those electrons from the glucose, and when it does so, it's going to make NADH. So we will yield some NADH through the process of glycolysis, and that NADH, since it is an electron carrier, it is going to hold on to those electrons temporarily, and then later, when we get to the last stage, it is going to contribute those electrons there. So that's the first stage. The second stage of cellular respiration is going to take place inside the matrix of the mitochondria. The matrix would be the innermost compartment of the mitochondria, and this second stage is called the citric acid cycle. This is at least one of the names of the stage. There are a lot of different names for this stage. It's also called the Krebs cycle. It's called the TCA cycle or tricarboxylic acid cycle. So it does have a lot of different names to it. This is the one we're going to try to stick with here. And it is a cycle. What will happen during this cycle is that the partially broken down glucose is going to get transported in, and it is going to be broken down even more. When it does get transported in, it will be converted into a molecule, or really two molecules, of acetyl-CoA. And then these acetyl-CoA are going to be broken down further. They will be broken down completely. So they will go around the cycle, and I'm just going to draw it like that. If we sum up over here exactly what the citric acid cycle is doing, it is going to complete the breakdown. And by complete the breakdown, really what we're saying here is that it's going to complete collecting the electrons. So complete electron collection. As it completes that electron collection, it is going to make even more NADH and also some FADH2, which is a very similar molecule. And those, again, are going to temporarily hang on to the electrons, those valuable electrons from the organic carbon. Lastly, what the citric acid cycle is going to do is it's going to make a little bit of ATP. There will be 2 of these, and they will also be by that substrate level phosphorylation process. Once we get to the third stage of cellular respiration, all of the carbon that we've been producing or breaking down along the way, it is gone, and it has left the cell, left the body as carbon dioxide. So what we're left with really is just the electrons that we've been collecting. So we have electrons out here. We have these guys, which are actually inside the matrix. So they're right here. And the third stage is going to occur right here on top of this membrane. This third stage is going to be called oxidative phosphorylation. This process has 2 different halves to it. Part A, or part 1, is called the electron transport chain. And so I'm going to write the electron transport chain down here. So this is part 1 of stage 3. We call it the ETC for the electron transport chain, and that will be molecules that are actually embedded in this inner membrane of the mitochondria. They will be embedded there. Their job is to extract the energy from the electrons, So I say extract, or you could say release the energy from the collected electrons. Again, those electrons are from the NADHs and FADH2s that we've been producing. And what they're going to do with that energy is they're going to create a hydrogen ion gradient. That gradient is going to be established across the inner membrane of the mitochondria. These little dots right here represent hydrogen ions. There will be a whole bunch of them in this intermembrane space right here, and there will be just a few or a low concentration inside the matrix of the mitochondria. So again, these are hydrogens that I'm drawing. Since the electron transport chain has created that big difference in hydrogens on the two sides of the membrane, that's going to serve as an energy source. So that when we get to stage number 2 of part 3, this is going to be chemiosmosis. Chemiosmosis is going to take advantage of that hydrogen ion gradient, and it's going to use it to make ATP. And we are going to make lots of ATP. It's really up to 36 total. That number does vary a little bit up and down because some of the hydrogens will get used for some other processes, but it is ultimately going to make a lot of ATP. The enzyme that's involved in this is called ATP synthase, and that enzyme is also embedded in this inner membrane. So I'm going to put it over here. So we can have one right here. I'll put another one right here. They would be all over this membrane. And what that ATP synthase is going to do is it's going to allow the hydrogens to flow back into the matrix, which is really exactly where they want to go because they want to diffuse down their concentration gradient. And as they do that, this ATP synthase is going to be taking ADP plus phosphate, and it's going to be combining them into ATP. And again, that is the big product of cellular respiration, is making lots of ATP using the energy that's released from the organic molecules that we've been collecting kind of all along the way.